Hi everyone. Now we are going to discuss about the glycolysis. What is this glycolysis? It is the conversion of glucose or glycogen into pyruvate or lactate with the production of ATP. So that is the sequence of reactions converting glucose or glycogen to pyruvate or lactate with the production of ATP. So we will see what happened inside the glycolysis. We will start with the glucose. This glucose will be converted into glucose 6-phosphate. This reaction will be carried out in presence of the enzyme hexokinase or glucokinase. What will happen? Here ATP will be converted into ADP. This reaction will be carried out in presence of magnesium ion. So we are converting the glucose into glucose 6-phosphate. The next step is glucose 6-phosphate will be converted into fructose 6-phosphate. This reaction is carried out in presence of the enzyme phosphohexose isomerase. The next step is fructose 6-phosphate will be converted into fructose 1,6 biphosphate. Here also ATP will be converted into ADP. This reaction is carried out in presence of magnesium ions. The enzyme that catalyzes the conversion of fructose 6-phosphate into fructose 1,6 diphosphate is phosphofructokinase. The next step is fructose 1,6 diphosphate will be converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This conversion of fructose 1,6 diphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate is catalyzed by the enzyme aldolase. So this is the interconvertible, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, that will be catalyzed by the enzyme phosphotriose isomerase. The next step is conversion of this glyceraldehyde 3-phosphate into 1,3 biphosphoglyceraldehyde or glycerate. 1,3 biphosphoglycerate. This reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Here NADH2 will be converted into NAD and phosphate ion will be eliminated. The next step is conversion of this 1,3 biphosphoglycerate into 3-phosphoglycerate. So we are converting this 1,3 biphosphoglycerate into 3-phosphoglycerate. This reaction is catalyzed by the enzyme phosphoglycerate kinase. What will happen here? Here ATP will be converted into ADP. This reaction is carried out in presence of magnesium ions. So this 3-phosphoglycerate will be converted into 2-phosphoglycerate. This is a interconvertible reaction. 3 will be the third position phosphate will be converted to second position. So 3-phosphoglycerate will be converted into 2-phosphoglycerate. The next step is formation of phosphoenol pyruvate. This reaction is catalyzed by the enzyme enolase. What will happen here? ADP will be converted into ATP. This is catalyzed by the enzyme enolase in presence of magnesium ion. This phosphoenol pyruvate is converted into pyruvate. Enol form will be present. This is catalyzed by the enzyme pyruvate kinase. So phosphoenol pyruvate will be converted into pyruvate and enol form will be formed. And spontaneous reaction, it will be converted into keto form. That is pyruvate keto form will be formed. So this pyruvate will be converted into lactate. This is catalyzed by the enzyme lactate dehydrogenase. Pyruvate keto form will be converted into lactate. This is catalyzed by the enzyme lactate dehydrogenase. This is the glycolysis, that is the conversion of glucose or glycogen into pyruvate or lactate with the production of ATP. When glucose is converted into pyruvate, if it is pyruvate is formed, it is called as aerobic reaction. Whereas in absence of that is in absence of oxygen, that is anaerobic reaction, that glucose or glycogen will be converted into lactate. So we can call it as conversion of glucose or glycogen into pyruvate or lactate, that is known as glycolysis. If it is aerobic, pyruvate will be the end end point. If it is anaerobic, then lactate will be the end point. I hope you could understand the sequence of reactions involved in the glycolysis.